All right, let's jump right in. Welcome to this deep dive, all about pharmaceutical analysis. You're in the right place if you're looking to really get a handle on the key terms and techniques, especially for your B.Pharm studies. And trust me, we're going to make sure you understand it all, not just memorize a bunch of definitions.
Yeah, exactly. We're going way beyond just passing exams here. Think of this deep dive as your foundation for a successful career in pharmacy, whether you're picturing yourself in a lab or working directly with patients.
So let's start with the basics. What exactly is pharmaceutical analysis?
It might sound intimidating, but really it's all about testing. We test everything from the raw materials to the final medications, like tablets, capsules, injections, even how those drugs act in the body.
And what's the goal of all this testing?
The goal is to make sure that every single medication that reaches a patient is safe, effective, and high quality.
Okay, so it's like a really detailed inspection process for medications
Yeah.
to guarantee they do what they should without any surprises.
Exactly. It's all about building trust.
Yeah.
When someone takes a medication, they're putting a lot of faith in the people who made and tested it, and that's where pharmaceutical analysis comes in.
I see. It's like a safety net for patients.
Mm.
So in our notes, I see this word assay popping up a lot. I'm assuming it doesn't involve knights in shining armor.
Haha. No jousting in the lab, I promise. An assay is basically a test that tells us how much of a specific substance is in a sample. Like imagine you have a paracetamol tablet, the pain reliever. The assay confirms it actually has the right amount of paracetamol, like the 500 milligrams it says on the label.
So it's like checking to make sure you're getting what you paid for.
Yeah.
No shortchanging on the paracetamol.
Exactly. And that's critical for getting the dosage right, which obviously has a huge impact on someone's health. Too little and the medication might not work. Too much, and you could have some serious side effects.
Makes sense. Dosage is definitely important.
Okay.
Next up, purity versus impurity. They seem pretty self-explanatory, but is there a specific nuance in pharmaceutical analysis?
Yeah, they're definitely two sides of the same coin. Purity means how much of the substance is the actual desired product, the pure active ingredient. Impurities are those little extra bits that can get in during manufacturing or even storage.
Okay, I think I get it. Is it sort of like baking a cake?
Yeah.
You want most of it to be flour, sugar, eggs, but sometimes a little eggshell sneaks in.
Perfect analogy. Some impurities are harmless, but others can interfere with the drug or cause unwanted side effects. Think about aspirin during production. You might end up with a bit of salicylic acid left over as an impurity.
And if there's too much of that salicylic acid.
It could irritate your stomach.
So impurities aren't always a bad thing, but controlling them is key.
Got it.
Now, two terms that always confuse me in school: precision and accuracy. Are they really that different?
They are, and understanding the difference is super important in our field. Picture throwing darts. Precision is hitting the same spot over and over, maybe on the outer ring. Accuracy is hitting that bullseye, even if your throws are a little scattered around it.
So I could be the most precise dart thrower, hitting the same spot on the board every time, but if it's not the bullseye, I'm not actually accurate.
Exactly. We need both in pharmaceutical analysis. A test can be precise and give you the same wrong answer every time, or it could be accurate overall, but with results all over the place. Neither is good.
So we need results that are both consistent and on target. Okay, buckle up. Here come two more. Sensitivity and specificity. Just the words make me a little nervous.
They're not as scary as they sound, I promise. Imagine a detective working a case. Sensitivity is like a bloodhound, able to pick up even the faintest scent. Specificity is like a fingerprint expert, identifying that one unique individual.
So a sensitive test finds what you're looking for, even if there's just a tiny bit. And a specific test is all about making sure you've got the right thing.
Right, and techniques like HPLC, high-performance liquid chromatography, are awesome because they're both. They can identify and measure drugs even in super complex mixtures. Think of it like picking out a single grain of rice from a bowl full of different grains.
Wow, HPLC sounds impressive. Before we get into more techniques, let's clarify two more: LOD and LOQ. What do they stand for again?
Limit of detection and limit of quantification. It's about understanding the limits of what we can measure. Imagine you're analyzing a drug that's barely there, in super low concentration. LOD is like a signal flare saying, hey, this drug is present.
But it doesn't tell you how much is there.
Right. That's where LOQ comes in. It's the point where you can confidently say, this drug is here and I know exactly how much of it there is.
Okay, LOD is like knowing someone's at the door, and LOQ is opening the door and seeing who it is. That makes sense. So we've talked about HPLC. What are some other key techniques in pharmaceutical analysis?
Well, there are tons, but let's start with one you might remember from high school chemistry: titration.
Titration. Ah, yes, lots of colorful liquids and swirling flasks. What was titration all about again?
It's all about controlled chemical reactions. We use a solution we know the concentration of to figure out the concentration of an unknown substance.
So we slowly add one solution to the other until something happens that tells us we're done.
Exactly. There's usually a visible change, like a color shift, that signals the endpoint. And from the amount of solution we added, we can calculate the unknown concentration.
It's all coming back to me now.
Yeah.
But how does this apply to pharmaceuticals? What are we actually measuring?
Titration is used for all sorts of things, like figuring out the purity of substances or analyzing the amounts of specific ingredients. Like acid-based titrations are commonly used to check the purity of citric acid in drug formulations.
So it's another quality control check, making sure the ingredients are up to snuff. Got it. What about chromatography? That one always sounds a little scary.
It might sound intimidating, but it's really just about separating things. Imagine you have a bowl of different colored candies. You could separate them by color, size, maybe even flavor. In chromatography, we separate the components of a mixture based on their chemical properties.
So instead of candies, we're separating different molecules in a drug sample.
Precisely. And while there are many types, in our world, we use HPLC a lot. Remember how we talked about sensitivity and specificity? Well, HPLC has both, making it perfect for identifying and quantifying drugs, even in very complex mixtures.
HPLC is like a super sleuth of the chemistry world.
Yeah.
I'm starting to understand why it's such a big deal.
It really is a powerful tool, and one common use is checking the purity of APIs, active pharmaceutical ingredients, which are the essential parts of any medication. By separating the API from any impurities, we can figure out how pure it is and make sure it meets those strict safety standards.
So HPLC helps make sure the active ingredient is actually active. Okay, let's shift gears a bit and talk about spectroscopy. Now that one sounds like something straight out of Star Trek.
Haha. Maybe not quite as futuristic as Star Trek, but we do use light to analyze matter. Just like light comes in a spectrum of colors, matter interacts with light in its own special way based on what it's made of. And spectroscopy lets us study those interactions.
So we're using light to unlock the secrets of molecules.
Exactly. There are different types of spectroscopy, but in pharmaceutical analysis, we use UV visible spectroscopy a lot.
UV visible. So ultraviolet and visible light.
You got it. Lots of drug molecules absorb light in the UV visible range. And by measuring how much light gets absorbed at different wavelengths, we can identify and quantify the drug.
So it's like shining a special light on the sample and seeing how much of it gets soaked up by the molecules.
That's a great way to put it. The more light that's absorbed, the higher the concentration of the drug. This helps us figure out things like the strength of a drug formulation or even how much drug is in a patient's blood.
Wow, that's incredible. It's amazing how we can learn so much from just shining light on things.
Mm-hmm.
So we've covered a lot of techniques, assays, titrations, chromatography, spectroscopy. How do they all fit together in the big picture of making medications?
That's a great question. All of these techniques are critical in the final stage. Quality control, or QC. Think of it like the last checkpoint before a medication can be sold.
Quality control, that sounds important. So this is where we make sure everything is good to go, right?
Exactly. Quality control ensures that every single batch of medication meets the required standards. We've talked about assays for potency, purity checks, and those super precise HPLC and spectroscopy measurements. All of these and many others are part of the big QC process.
So it's like a huge system of checks and balances to guarantee that the medications people take are safe, effective, and high quality. It makes me feel a lot better knowing so much effort goes into making sure each pill or capsule is exactly what it's supposed to be.
It's a rigorous process for sure, but it's absolutely necessary. Patient safety is paramount, and pharmaceutical analysis is woven into every step, from the initial analysis of raw materials to the final product testing. We've covered a ton in this first part of our deep dive. Ready for a little break before we uncover even more about this fascinating field?
Absolutely. Let's take a quick pause and then dive right back into the world of pharmaceutical analysis.